All right, so this question looks at lethal alleles, and it's from, I believe, the 2015 Level 2 genetics paper. So the question is, in 1905, Cunot observed unusual ratios when studying inheritance of coat color in mice. After mating two, and let's go through and look at these important bits of information that they are giving us. So two heterozygous yellow mice, so they would have had the genotype big Y, little y. He observed that the offspring never showed a normal 3:1 phenotypic ratio. Instead, he always observed a 2:1 ratio. So straight away, you should be thinking lethal alleles whenever you see the 2:1 ratio. So with two yellow mice for every grey mouse, he concluded that yellow coat colour, big Y, was dominant over grey coat colour, little y. And by using test crosses, he showed that all his yellow mice were heterozygous. However, from his many crosses, Cunot never produced a single homozygous dominant yellow mouse. Subsequently, it was confirmed that no homozygous dominant yellow mice were present because of a lethal allele. So in this one, they're kind of nice. They actually give you a bit of an idea that we are talking about lethal alleles. First question here is describe a lethal allele. So I would say something along the lines of the fact that causes death when you have the specific genotype. And the genotype could be homozygous dominant, homozygous recessive, or it could be heterozygous. Okay, so it is whatever genotype is lethal. Any individual that receives that genotype will die, whether that is before birth because development is affected, or whether just after birth because for some reason that lethal genotype combination means that that individual just can't survive. It does not have the means to live. So the next question is discuss how Cunot used test crosses to determine that all the live yellow mice were heterozygous. In your answer include a description of homozygous and heterozygous. Let's go through and highlight some of these key points again that they're giving us. So they want us to talk about homozygous and heterozygous, an explanation of what a test cross is, and a discussion of how Cunot used the test crosses to observe a 2:1 ratio and determine that all live yellow mice, I should say, were heterozygous. Okay. So let's start off with, all right, so homozygous. So homozygous, you would say, is when you've got two the same. So homozygous, the beginning there, homo meaning same. I'm just using R's as an example. Or it could be little r, little r. This is obviously homozygous dominant, and this is homozygous recessive. Heterozygous, and again, it's in the beginning there, hetero, meaning different. Okay. So your homozygous is where you've got two of the same genotypes. So it's two of the same alleles. And heterozygous is where you've got two different alleles. Okay, one of them is going to be dominant and one of them is going to be recessive. You could go on to say that in a heterozygous individual, you only see the dominant trait coming out. An explanation of what a test cross is. So a test cross is where you've got an unknown individual genotype. So you're not sure whether they are homozygous dominant or homozygous recessive, and you want to find out what they are. And by doing that, you would mate them against a wild type individual. So that might be the homozygous recessive, it could be the homozygous dominant. Okay, so how did Kennard use the test crosses to observe a 2-to-1 ratio? So let's think again back to his first original cross. So he crossed an individual like this. This individual would have died, so we would have ended up with 2 of those and 1 of those. And our phenotype ratio would have been 2 yellow, 1 grey. A way to prove that it is this individual that is living and not big Y individual. Let's do some test crosses. So, say the individuals that were surviving were actually homozygous dominant. We would make them in a test cross with an individual that was homozygous recessive. So this would be a grey individual. What we would find, if it was in fact the homozygous dominant individual that was surviving, and we crossed them with a grey individual, all these offspring would die. Okay. But what he found was then that when he was mating these individuals, he was getting some yellow and some grey. So if we did another test cross, oops. That would be a little y. He was getting grey individuals being born. So doing a test cross with those living individuals against a homozygous recessive, so a grey individual, he would find that he would get all living individuals, and you would expect to see two lots of yellow to 2 lots of grey. So doing that test cross, we can show that the yellow individuals who are living must be heterozygous, big Y, little y. Because if they were in fact big Y, and we mated them with a grey homozygous recessive individual, we would get no live offspring. But because when he did breed them together in a test cross, a grey and the yellow individuals who are living, he would find that he would get 2 lots of grey and 2 lots of yellow, showing that the living individuals have to be heterozygous. I'm hoping that answered all of that part there. Let's get rid of some of this. Okay, moving on to the next question. So this was actually quite a long question, really. C. The genetic disease cystic fibrosis is caused by lethal alleles. An infected individual is homozygous recessive. However, heterozygous individuals are carriers of the lethal allele. Lethal alleles are caused by mutations. The mutation of cystic fibrosis occurs in the gametes. Discuss how mutations cause lethal alleles and why cystic fibrosis alleles remain in the population. So what are they wanting us to do? They're wanting us to describe what a mutation is. Whenever you see describe, we're talking achieved. They want an explanation of the difference between a gametic mutation and a somatic mutation. Whenever you see explanation, they're talking mirrored. A discussion is always excellent. Discussion of why the cystic fibrosis lethal allele remains in the human population. Now, cystic fibrosis is a tricky one because, yes, it is an example of a lethal allele in humans, but just because you have cystic fibrosis does not mean you die at birth. Individuals with cystic fibrosis can live now because of medication into their 30s. Used to be kind of late teens, early 20s. Now these individuals can live into their 30s. So a mutation is usually a change in the DNA, and that might be the bases. Okay, you might have an A change to a C. Okay, it might be whole chromosomes. Okay, bits of chromosomes mixed up and shuffled and where they're not meant to be, deleted completely, duplicated, that kind of thing. So mutation is a change in the DNA, and it usually causes some sort of difference in the phenotype. So let's look at the next part, an explanation of the difference between a geometic mutation and a somatic mutation. So mutations can be caused from the environment, chemicals, UV, especially important to us with melanoma. So a somatic mutation occurs in your normal cells, normal body cells. A gametic mutation will be one that occurs in the sperm or the eggs. Okay. A good example of preventing some gametic mutations for boys is when you have an X-ray, you might be asked to put a lead apron over your testicles to prevent any sperm from being mutated because of the X-rays that they're using. So if a mutation occurs in a normal body cell, it won't be passed on. It'll affect maybe that body cell and body cells around it. If it occurs in the sperm or the egg, it is passed on to the next generation. Okay, and finally, a discussion of why cystic fibrosis lethal alleles remain in the human population. With this one, because it can just be a random mutation, we can't stop mutations from happening. So if a mutation happens, and it happens is a gametic one, happens in the egg or the sperm, it's going to be passed on. If that egg or sperm then go on to make another individual, it's going to be passed on to the next generation. If, I guess one way we could check is to do testing before individuals are born. Cystic fibrosis, I'm not sure if it can be picked up on a scan. Obviously, it would need to be genetic testing. That would be the only way to stop it from being in the human population, was to, I guess, get rid of those individuals with cystic fibrosis before they are born. But if it is coming from a mutation, we cannot stop mutations from occurring in the gametes. So I hope that's helped. Have a look through this question yourself. And give it a go. As always, I am happy to mark any work that you have. Thanks.